Hello students, welcome to the new session of Cell the Unit of Life. Okay, so today we are going to solve the exercise, NCERT exercise of the chapter, the Cell the Unit of Life. Okay, so let us start with the first question here. The first question says, which of the following is not correct? Okay, so there are four options. Option A, Robert Brown discovered the cell. Option B says Schleiden and Schwann formulated the cell theory, and Virchow explained the cells are formed from pre-existing cells. This is the option C. And option D says a unicellular organism carries out its life activities within a single cell. So out of this, which of the following statement is incorrect, right? So let us see the answer of this. Okay, so the answer is Robert Brown did not discover the cell. The cell was discovered by Robert Hooke. Okay, so all the three statements here, except the A, that is Schleiden and Schwann formulated the cell theory. This is absolutely correct. The cell theory was given by Schleiden and Schwann, right? And later the cell theory got you can say that cell theory got a new phase, that this thing was formulated by the Virchow and it was added to the new cell theory, right? That Virchow explained the cells are formed from the pre-existing cell. New cells are coming, right? And from where these new cells are coming? These new cells are coming from the pre-existing cells. Okay, so option C is absolutely correct. Now coming to the option D, a unicellular organism carried out its life activities within a single cell. So all the life activities, whether it is a nutrition, whether it is excretion, whether it is a respiration, okay, and if it is a plant cell, then photosynthesis, everything, all the metabolic activities, all the life processes, okay, in a single cell organism, right? It is occurring inside the cell only. So the statement a unicellular organisms carries out its life activities within a single cell, right? So this is also an absolutely correct statement. Coming to the statement 1, Robert Brown discovered the cell theory. Robert Brown discovered the cell. Now this statement is, the Robert is okay, but the Robert Brown discovered the nucleus, right? He saw first time the nucleus, but Robert Hooke was the person who first time discovered the cell. He cut the slice of the cork cell and see under the his own handmade microscope, and in that he saw the honeycomb like a structure, and to that he gave the name cell, right? The space that is there, so he gave that a space as a cell, right? So Robert Hooke discovered the cell, not Robert Brown. Okay, now coming to the next question here. The new cell generate from, okay, the new cell generate from, option A says bacterial fermentation. New cell generate from bacterial fermentation. Option A is bacterial fermentation. Option B, regeneration of old cell. Okay, regeneration of the old cell. C option says pre-existing cells, and D says abiotic material. So the new cell generate from bacterial fermentation. Every cell's coming from the bacterial fermentation? No, it's not correct. Okay, so what is the correct? According to the biogenetic theory, the new cells can only arise from the pre-existing cells, and this was formulated in the cell theory, right? Cell theory was given by Schleiden and Schwann, right? But the concept that the new cells are arising from the pre-existing cell came from the Virchow, and that was added in the new cell theory. right? So according to the biogenetic theory, new cells can only arise from the pre-existing cell, and only the complete cells in favorable condition can give rise to the new cell. So when the favorable condition is very favorable for the growth of the plants, then what happen? These cells forms the new next generation cells, right? So looking over the options here, the new cell generate from the option A, bacterial fermentation. This is absolutely incorrect. Regeneration of old cell. We are talking about the new cells. Now old cell get regenerated to form the new cells. Regeneration is seen in some of the organism, not in every organism, and every cell is not having the power of regeneration. So this is also an incorrect statement. The pre-existing cell, this is the correct option. And new cell generate from the abiotic materials. No, abiotic materials, that is not living. So new cell generate from the pre-existing cells, that is the living. So option D is also the incorrect, right? Okay. Moving to the next question. So this is the match the column type of question. Okay. So here it is based on the organelle structure, right? Sorry. So they are talking about the three different organelles: mitochondria, that is the Golgi apparatus, and the chloroplast, right? So let us see column 1. The option A is showing the Christie, and in the column 2, it is the first says flat membranous sacs and stroma. Now option B, cisternae, enfolding in the mitochondria. Option C, thylakoids, and third says disc-shaped sacs in the Golgi apparatus. So talking about the first one, that is the Christie. So Christie is what? We know that the mitochondria is a double membrane organelle. It is a double membrane organelle, and its inner membrane shows the invagination. Inner membrane shows the invagination and forms the Christie, and form the Christie-like structure. And on these Christie, only small tennis-shaped F1 particles are there. Tennis-shaped F1 particles are there, that is attached to the surface of the Christie. Now Christie increases the surface area, and these F1 particles that is attached to the Christie causes what? It is the primary function of this is to generate the ATP, right? So the Christie increases the surface area, so more number of F1 particles get attached to it, right? So the more number of ATP that can be generated. So the Christie, that is the enfolding in mitochondria. Now talking about the cisternae. Cisternae. So cisternae, where it is present? Cisternae is present in the disc-shaped Golgi apparatus. If you see the Golgi apparatus structure, then you can see the disc-shaped structure, flattened structure, parallel to each other, and their number can be 4 to 10 or 12 even. And these flattened structures are called as cisternae. So these are the disc-shaped sacs in the Golgi apparatus, right? So that is called as cisternae. So B is matching with the 3. Option C, that is the thylakoids. So thylakoids is a flat membranous sac that is present in the stromata. Stroma. Stroma is the liquid substance that is present inside the chloroplast, right? And again, this if you're talking about the chloroplast, then it is a double membrane structure, and inside the double membrane structure, what you see there are some flat structures that are called as thylakoids. Now there are small thylakoids which are piled one above the other, and that is called as grana. So there are number of grana, and the granas are attached with some stroma lamellae. or the stroma thylakoids. So basically the thylakoids are the flat membranous sacs in the stroma, and on the surface of these thylakoids only, what is attached? Chlorophyll molecules are attached, right? Chlorophyll A, that can be chlorophyll A, that can be chlorophyll B, right? But this chlorophyll are attached to this flat membranous sacs that is present in the stroma, and that is called as thylakoids. So looking over the match, the correct match we can see here, cisternae, that is showing the enfoldings in the mitochondria, Christi, that is showing the enfoldings in mitochondria, cisternae, that is the disc-shaped sacs that is present in the Golgi apparatus. If you see the Golgi apparatus, there are the thin disc-shaped sacs, okay, that is called as cisternae. Thylakoids. Thylakoids are the flat membranous sacs that is present in the stroma, the liquid substance that is present in the chloroplast. Okay. Okay. So this is showing the correct match. Now moving to the next question. Which of the following is correct? So there are the 4 different statements. One of the statement is correct, whereas the 3 are incorrect. You have to choose the correct one. So let us read the statement first. A, cells of all living organisms have a nucleus. Every, okay, cells of all living organisms, there is a nucleus. Both animal and plant cell have a well-defined cell wall. Option C, in prokaryotes, there is no membrane-bound organelles. Option D, cells are formed de novo from the abiotic material. Okay, so if you see the correct answer, then the correct answer is the C1. The membrane-bound organelles are the organelles surrounded by the double membrane. So nucleus, mitochondria, chloroplast are the example of such organelles. These cell organelles are totally absent in the prokaryote. So option C is correct. Now if you see, the cells of all living organisms have a nucleus. Every living organism have a nucleus. Okay, cells of all living organism. Talking about the RBC, okay, they are not having any nucleus in that. And also if you see the prokaryotic cell, there is no true nucleus. So this statement is not fully correct, right? Both animal and plant cell have a well-defined cell wall. Cell wall is only present in the plant cell, that is not present in the animal cell. So this statement is also not correct. The correct one is the C. We have already discussed, in prokaryotes there are no membrane bound organelles. The membrane, true membrane, that is a true membrane means a lipid bilayer, and that is traversed by the protein, right? So that is showing the plasma membrane structure. So that is a true membrane. If any membrane that is not like the plasma membrane, that is not considered as a true membrane. But in the prokaryotes, there is no true membrane bound organelles. So that is clear. Okay. Option D, cells are formed de novo from the abiotic materials. No, the cells are formed from the pre-existing cells. So this statement is also incorrect. Only C option, in prokaryote there is a no membrane bound organelles. This is the true statement. Okay. Okay. Moving to the next question. What is mesosome in prokaryotic cell? Mention the function that it performs. So we have to tell about the mesosome. So we all know that the bacterial cell or the prokaryotic cells, they are having membrane bound, right? They are having the membrane bound, and at some places what happened, their membrane shows the invagination. There is a lot of invagination and they get enfolded. This membrane get at one certain points, they get enfolded, right? And that is forming the mesosome structure. And this you see in the case of prokaryotic cell only, right? So mesosome is a convoluted membranous structure formed in a prokaryotic cell by the invagination of the plasma membrane. So this is the invagination of the plasma membrane. They have the plasma membrane. That plasma membrane get highly invaginate and form a coiled structure like this. And its functions are as follows. Now they are having certain functions. So let us see what are the functions of the mesosome. So these extensions help in the synthesis of the cell wall, replication of DNA. Replication of DNA, that DNA makes its own copy. Okay. So in that also, this mesosome is going to help this DNA structure to get replicated or duplicated or make its own copy. So how it happened? The plasma, this extension that is called as a mesosome, at its surface, the DNA, the circular DNA, that is the naked DNA, right? That get attached and it is helping to replicate it. Now these extensions help in the synthesis of cell wall in the case of prokaryotes. They also help in equal distribution of chromosome in the daughter cells. Now when the DNA or the circular DNA, that is the circular chromosome, get duplicated, then each cell get equal distribution of the chromosome with the help of this mesosome. So in that also it is going to help the cell. It also increases the surface area of the plasma membrane to carry out various enzymatic activities. Now this is going to be a very, very important point, right? Now mesosome is having all those enzymes at its surface that is helping the process of cellular respiration. Whatever the respiration process takes place in the mitochondria, in the mitochondria, that is in the eukaryotic cells, they are present, and they are the primary function of the mitochondria is to do the process of cellular respiration. So here the cellular respiration process is taking place at the surface of mesosome. Why? Because they are having all those enzymes at its surface, right? So it also increases the surface area by unfolding. It increases the surface area. So more and more enzymatic activities that can take place at its surface, right? And what enzymatic activities? The enzymatic activities that are involved in the process of cellular respiration. And it helps in secretion process as well as in the bacterial respiration. So all these are the function of the mesosome, right? Again, mesosome is the invagination of the plasma membrane. So you do have to remember this. This is a very, very important point, and this is a very important question as well. Okay, so moving to our next question. How do neutral solutes move across the plasma membrane? So neutral solutes, that is not having any charge over it, positive charge or negative charge, polar or non-polar, it is a neutral, right? So how this neutral solutes are moving across the plasma membrane? The question is about that. Okay, they are having another question in that only. Can the polar molecules also move across it in the same way? And if not, then how are these transported across the membrane? So moving to this answer of this question. Okay, so it talks about plasma membrane is the outermost covering of the cell that separated from the environment. Now this is going to be a very important point. This plasma membrane is made up of a lipid bilayer and the protein are traversed in between. Okay, so there are two different types of protein: intrinsic protein that is present between the bilayer and extrinsic protein at the surface, right? So they are the outermost covering of the organism and that's separated from the outer environment. Second point, it regulates the movement of the substances into the cell and out of the cell. Whatever the substance is coming inside or outside, it is coming from this membrane only or moving out of this membrane only from the plasma membrane, and it allows the entry of only some substances and prevent the movement of the other materials, right? So it means that it is selectively, it is allowing only some of the substances to move inside the cell and preventing some of the substances that are not allowed to come inside the cell, but it is allowing only the sums of the substances that is required by the cell. So selectively it is allowing some of the substances to enter inside the cell, right? And some of the substances are allowed to move out of the cells also. It means that the plasma membrane is selectively permeable, right? Movement of neutral solute across the cell membrane. Neutral molecules move across the plasma membrane by the simple passive diffusion. Passive diffusion. According to the concentration gradient, it is not having any type of, we can say, charge molecules over it, right? So diffusion is the movement of the molecules from the region of high concentration to the region of the lower concentration. So you have studied about the diffusion. Diffusion means, say, diffusion says what? Diffusion says that if any particles that are in larger concentration, so it will move from higher concentration to the region of lower concentration. Now here again the movement of the neutral molecules are occurring in the same way through a semi-permeable membrane. So there is no selectivity. Okay. Movement of polar molecules across the cell membrane. Now we will talk about how the polar molecules are moving across the cell membrane. The cell membrane is made up of phospholipid, bilayer and protein. Now this is very important. The cell membrane is made up of phospholipid. This is a type of a lipid by which this bilayer lipid layer that is present in the plasma membrane is made up of, and some proteins also there. We have already discussed about it. The movement of the polar molecule across the non-polar lipid bilayer requires carrier protein. So how this polar molecules are moving across the cell, they require the carrier protein. Now the protein, intrinsic protein or extrinsic protein we are talking about, these are there to allow the polar molecules to move across the plasma membrane. So the carrier proteins are integral proteins. Integral proteins means it is traverse the lipid bilayer, right? And it is the integral protein particles having a certain affinity for a specific solute. So they are not having the specificity for every type of solutes. For certain specific solute get attached to it, and it allows the particles to move across the plasma membrane. And as a result, they facilitate the transport of molecule across the membrane. So in this way, this lipid, we have talked about the lipid molecule, that is a lipid bilayer is there. There is a intrinsic protein. So you now understand what is the function of these intrinsic protein. Now there are functions of this lipid bilayer also. There are certain substances that is made up of lipid. They can get easily across the lipid bilayer, right? There are certain hormones, okay, that are made up of lipids. So they can enter inside the cell easily through the bilayer of the lipid. But the polar molecules, how they are getting entered inside it, for that there are specific carrier proteins. Where these carrier proteins are coming about? These carrier proteins are the same protein, that is we are talking about integral protein. That is the carrier protein. Okay. So polar molecules are moving across the plasma membrane through the carrier proteins. Non-polar molecules or the neutral molecules, they are moving across the plasma membrane through the simple diffusion process. And In this question, only these two things about asked. But if sometimes it is asked about how the hormones are moving, so hormones are basically, they are made up of the lipid, then it can move easily through the lipid bilayer, right? Next question comes here. Name two cell organelles that are double membrane. So there are different types of organelles that are present inside the cells. There are some organelles that are double membrane. There are some organelles that are single membrane, right? So here it is a double membrane bound organelle. They are talking about the two. You have to discuss about the two cell organelles that are double membrane bound, and what are the characteristics there's two organelles. You have to tell about the characteristics of those two double membrane cell organelles. Now state their function and draw the label diagram of both. So let us see the answer. Okay, so the first two organelles, mitochondria and chloroplast, are the two organelles that are double membrane bound, right? So let us see the diagram. Now here you can see the diagram of the mitochondria, a double membrane. You can see the inner membrane and outer membrane, and there is a, in between these two membrane, there is an inner membrane space. Now the inner membrane get invaginated, that is forming the cristae, and at the surface of the cristae there are F1 particles. Okay, F1 particles, and these F1 particles are actually ATP synthase particles. ATP synthase particle. Okay, and the liquid substance that is present inside the mitochondria, that is called as matrix, and there are some many granules are also present. So let us talk about the characteristics of mitochondria. Mitochondria are the double membrane bound structure. So it is a double membrane bound structure, outer membrane and inner membrane. The membrane of the mitochondria is divided into the inner and outer membrane, distinctively divided into two aqueous compartments. So they are making two compartments. The compartment that is present in between the outer membrane and inner membrane, and the compartment that is formed by the inner membrane itself. Okay, so two aqueous compartment, outer and inner compartment. The outer membrane is a very porous, containing the organelle. Okay, it is quite porous, so substances can move easily, while the inner membrane is deeply folded. It is highly folded structure that is forming the cristae, that is forming the structure cristae. These folds are known as cristae. Okay, the inner membrane that invaginate and forming a infolding, that is called as a cristae. Now cristae increase the surface area inside the cell. It increases the surface area inside the cell, and they are the site of ATP generation at this particular point only, right? And the ATP get generated. There are a lot of chemical reactions taking place over there, and the energy that is released, it is stored in the form of high energy phosphate bond that is present in the ATP molecule. The membrane of a mitochondrion contains a specific enzyme meant for the specific mitochondrial function. Now the membrane, there are two membranes. Each membrane is having its own specific function. So that is why it is written here. A mitochondrion contains a specific enzyme meant for the specific mitochondrial functions. Hence, the mitochondria are the site of aerobic respiration. Aerobic respiration, it means that it requires oxygen. They have their own DNA and ribosome. Very, very important point. The mitochondria is having its own circular DNA, and they are having the ribosomes also, and these ribosomes are of 70S type that you see in the case of prokaryotic cell. Same type of ribosomes are present in the mitochondria as well. Thus, they are able to make their own proteins. These are able to make their own proteins, and this is why they are considered as semi-autonomous organelles. They can divide and redivide and forms the new mitochondria, right? So that is why it is called as semi-autonomous organelles. So this is about the mitochondria. Now let us discuss about the another double membrane organelle, that is called as chloroplast. So chloroplast is a double membrane bound structure, and they are divided again. They will form the two compartment. The compartment that is present in between the outer membrane and inner membrane, and the compartment that is formed by the inner membrane. So they are divided into outer and inner membrane, further divided into two distinct regions. Inside the inner compartment, what you see, there are again the two regions. One region that is called as grana. Okay, grana, and they are the stacks of a flattened disc containing chlorophyll molecules. So grana are the thylakoid membrane that is stacked one above the other, and it is a pile of thylakoid membrane. A pile of thylakoid membrane. It is something like that you have placed a coin one above the other and forms a pile of coin, right? So the flattened membrane sacs are called as thylakoids, and the thylakoids of adjacent grana are connected by the membranous tubules called as stroma lamellae. So there are not a single grana, there are a number of grana, and granas are interconnected by the stroma lamellae. Stroma, what is then stroma? Stroma is a homogeneous mixture in which the grana are embedded. It is a liquid substance. It is a matrix substance of the chloroplast in which this grana are embedded. So it is a homogeneous mixture in which the grana are placed or embedded. It contains several enzymes that are used for the synthesis of carbohydrates and proteins. So this is very important, that this stroma part, it is having a lot many enzymes that are involved in the process of synthesis of carbohydrates and proteins, and it is also contain its own DNA and ribosome, as we have seen in the case of mitochondria. So in the same way, the chloroplast is also having its own circular DNA, and it is also having its own ribosomes, and these ribosomes are also of 70S type that we have seen in the case of prokaryotic cell. So both the mitochondria and chloroplast are having 70S type of ribosomes. Okay, so it also contains its own DNA and ribosomes. Now, function of mitochondria. We have discussed about the structure. Now let us discuss about the functions of mitochondria. So they are the site of cellular respiration. Mitochondria are the site where the cellular respiration takes place. They provide energy in the form of ATP for all vital activities of living cells. So ATP generation takes place in the mitochondria only at the surface of the cristae, because the F1 particles are attached to it, and the ATP synthase enzyme, that is the F1 particles. So they generate this ATP molecules. They have their own DNA and ribosomes. Okay, they are having their own DNA and their own ribosomes. Hence, they are regarded as a semi-autonomous organelles. So this is about the function of the DNA. Another function, that they have several enzymes intermediately required for the synthesis of various chemicals such as fatty acids, steroids, and amino acids. So that are also get synthesized in this particular mitochondria. Now let us discuss about the functions of chloroplast. What is the function of chloroplast? So they trap solar energy and utilize it for manufacturing food for the plants. Okay, so chloroplast, they are very important. They trap the solar energy, sun energy, and utilize it for the manufacturing of food. It means that light energy get converted into the chemical energy. Foods are in the form of chemicals, right? So the light energy that is coming from the sun, it gets trapped by the chlorophyll molecules that are present inside the chloroplast, and they convert it into chemical energy, food. Hence, they are involved in the process of photosynthesis. So this is the main, this pigments are the main component of the process of photosynthesis. If these pigments are not present, then the photosynthesis process cannot take place. Now, they contain the enzyme that is required for the synthesis of carbohydrates and proteins, right? So there are many different enzymes that are required for the synthesis of carbohydrates and proteins that are present inside the chloroplast region stroma. Coming to the next question, what are the characteristics of prokaryotic cell? We are having the two different types of cells, prokaryotic cells and eukaryotic cells. So here it is in the question that what are the characteristics of the prokaryotic cell. So let us see. Prokaryotic cell is a unicellular organism lacking membrane-bound organelles. The number one point, that what are the prokaryotic cells? The prokaryotic cells are the cells in a unicellular organism. It is a unicellular organism that is lacking membrane-bound organelle. Not a single membrane-bound organelles are present in the case of prokaryotic cell. Membrane bound, it means that true membrane. The true membrane is the plasma membrane. That type of membrane, if it is present, then we can say that it is a membrane bound, right? So that type of membrane is not present. The characteristic of the prokaryotic cells are as follows. So let us see what are the different characteristics. Number one, most of them are unicellular. Prokaryotic cells are all unicellular. They are generally small in size, and their size is very small, ranging from 0.5 to 5 micrometer. So this is the size of the prokaryotic cell, that is ranging from 0.5 to 5 micrometer. They are very, very small. So this is the size of the prokaryotic cell. Next, the nuclear region of the prokaryotic cell is poorly defined because of the absence of nuclear membrane. There is no nuclear membrane is present in the case of prokaryotic cell. Hence, a prokaryotic cell lacks a true nucleus. It is not having any single nucleus, right? So they are having no true nucleus. Instead of it, it is having nucleoid structure. Okay, so there is a region in which the circular DNA is present, and it is not bound by any membrane like structure. So there is no true nucleus. Okay, and that is why it got its name prokaryote. Pro means primitive and karyo means, karyotes means nucleus. Primitive type of nucleus. Primitive type of nucleus is the nucleus that is not bound by any membrane like structure. The genetic materials of the prokaryotic cells are naked. And if you see what is the genetic material? It is a circular DNA like structure. It is naked. It is not covered by any membrane. They contain a single circular chromosome. That's it. And in addition to the genomic DNA, they have a small circular plasmid DNA. Now this is the circular DNA which is present in the center, whereas there are some circular DNA, small circular DNA, that are also present in the cytoplasm of the prokaryotic cell. And what its name? It is called as plasmid DNA. So they have a specialized membranous structures now called mesosome. Now, once we have said that they do not have any membrane bound structure, now here we are saying that they have a specialized membrane structure. Now this structure is not a unit. It is a single, at a point the plasma membrane get invaginated and it forms a mesosome like structure. So that is not another organelle type of things. It is just a single, at a single point, the membrane, the plasma membrane get invaginated and it is forming a highly, you can say that invaginated membrane that is called as a mesosome. Mesosome are formed by the invagination of the same cell membrane. So this is just the extension of the plasma membrane, right? It is not any membrane bound organelle. So these extensions help in the synthesis of cell wall and replication of DNA. So this is the point where the extension, you see, it helps in the cell wall formation, replication of DNA as well. They are also help in equal distribution of the chromosome into the daughter cell at the time of cell division. So when there is a replication of DNA, the DNA made its own copy. The two circular DNA now at the time of cellular division, okay, equal distribution of chromosomes is there. So that is also taken care by the help of mesosomes. Next point, membrane-bound cell organelles such as mitochondria, plastids, and endoplasmic reticulum are totally absent in the case of prokaryote. These are the true membrane-bound cell organelle which is present in the eukaryotic cell. But all these membrane-bound organelles are totally absent in the prokaryotic cell. Most prokaryotic cells contain three-layered structure. Outermost glycocalyx. Now if we are talking about the cell envelope, okay, so outer membrane, that is called as glycocalyx, middle cell wall, and the innermost plasma membrane. This structure acts as a protective unit and it is called as the cell envelope. Now the glycocalyx, it can be of two types. In some it is a slime layer, okay, very slimy layer, it's a very thin, and in some it forms the capsule like part, hard covering. So glycocalyx can be of two different types depending upon the type of prokaryotic cell or the bacteria. So examples of prokaryotic cell include the blue-green algae bacteria, blue-green algae, that is the cyanobacteria that contain the chlorophyll in it and do the process of photosynthesis, and bacteria, that is the normal eubacteria we are talking about. Moving to the next question, multicellular organism have division of labor, explain. So there are unicellular organism, there are multicellular organism. So here the question is about the multicellular organism and they are having a division of labor. You have to explain this. So let us see the answer. Multicellular organisms are made up of millions and trillions of cells. If you see any multicellular organism, it is having a number of cells that unite together to form a unit body, and it is made up of millions and trillions of cells. So all these cells perform a specific function. These cells unite together and perform a specific functions, and all these cells specialized for performing a similar functions that are grouped together as a tissue in the body. So they are performing a similar function, they are grouped together as a tissue in the body. Hence a particular function is carried out by a group of cells at a definite place in the body. Similarly, different functions are carried out by the different groups of cells in an organism, and this is known as a division of labor in the multicellular organism. In the multicellular organism, you can see at the tissue level of organization, you can see organ level of organization, and you can see organ system level of organization. So these three different. So there are different tissues. Every tissue performs the different functions. So here we can see according to the tissue, there is a labor of division. Okay, this tissue has to perform this function. Second type of tissue has performed the second function. Similarly, if you see the organ level of organization, then also you can see the particular organ is there to perform a particular function. Another organ perform another function, right? So again here in the organ level of organization, we the different functions. If we see the organ system of organization, then you see there is a digestive system that is only confined to do the process of digestion. Excretory system, it will perform the process of excretion. Circulatory system, right? Reproductive system, every system is performing a particular type of function. So this is what? This is a division of labor, right? So we see in multicellular organism the different division. Even in the single cell organism, unicellular organism, you see the division of labor. There are different organelles that are taking a particular functions. Okay, moving to the next question. Cell is the basic unit of life. Discuss in a brief. So cells are the basic unit of life capable of doing all the required biochemical processes that a normal cell has to do in order to live, right? So what is the basic function of a cell? The basic unit, cell is the basic structural and functional unit, and it required all the biochemical process that takes place inside the cell only. And why that is required? To live, in order to live. So all those biochemical process that is occurring inside the cell, right? They are the base for living a particular organism. So the basic need for the survival of all living organisms are the same. Whether it is a plant cell, whether it is an animal cell, whether it is a unicellular organism or a multicellular organism, the basic requirement of all the living organisms are the same. All living organisms need to respire, digest food for obtaining energy and get rid of the metabolic waste material that is synthesized during the course of the digestion of food, the respiration of food, right? So respiration is taking place in every cell, whether it is a single cell or a multicellular organism, right? Digestion is also occurring in every cell. So this is the thing that makes the basic unit of life. Cells are capable of performing all the metabolic functions in the body. Hence, cells are called as a functional unit of life. Moving to the next question, what are nuclear pores? State their function. Very, very important. So nuclear pores are present where? We have seen that there in the true nucleus, that is in eukaryotic cell, there is a nuclear membrane, and in between this membrane there are some pore-like structures, pores, okay, holes through which the component can go outside the nucleus and some can move inside the nucleus. So nuclear pores are the tiny holes present in the nuclear membrane of the nucleus. They are formed by the fusion of two nuclear membranes, right? So there are two nuclear membrane, outer membrane and inner membrane. So they forms the pore-like structure. And these holes allow the specific substances to be transferred into a cell and out from it. So it is making a compartment. There is a two compartment. Nucleus is making its own compartment, and the cytoplasm in between the plasma membrane and the nuclear membrane, there is one more compartment, right? So the substances is moving from the outer compartment to the inner compartment, that is the nucleus. And from the nucleus, it is moving to the cytoplasm also. So they allow the molecules such as RNA and proteins to move in both the directions. So where the RNA gets synthesized? RNA gets synthesized on the template of DNA inside the nucleus, and that is the process of transcription. So when the RNA is synthesized, it has to be transported outside the nucleus in the cytoplasm, because the ribosomes are there, and there that is the site for the protein synthesis, right? So they allow molecules such as RNA and proteins to move in both the direction between the nucleus and the cytoplasm. Both lysosome and vacuoles are the endomembrane structure, yet they differ in their terms of their functions. Comment. So lysosomes are the membrane-bound vesicular structures holding a variety of enzymes such as lipases, proteases, and carbohydrates. So these are the enzymes that are present in the lysosomes. Now the purpose of the lysosome is to digest worn out cells. Lysosomes are there, whatever the worn out cells or the damage of the cells, they just engulf that, and they are involved in the intercellular digestion of the foreign food particles and microbes. Sometimes the cells takes the foreign particles inside it, so all those particles are taken and they get degraded by these lysosomes. So sometimes they also act as a suicidal bed. Sometimes the whole cell get damaged. So these lysosomes can engulf and digest whole cell, and that is why it is also called as suicidal bags. They are involved in the self-digestion of a cell, and this self-digestion occurs just to clear out that damaged cells. So they are a kind of waste disposal system of a cell. So they are taking care of whatever the waste that is present or coming out of into the tissue by the damage of the cells or any other things, so they can clear out that things. And on the other hand, vacuoles are the storage sacs found in the cell. Whatever the extra substances that is there, they get stored inside the vacuole. Though they might store the waste product of the cells in unicellular organisms, the food vacuole contain the consumed food particles. It also play a role in expelling excess water and some waste from the cell. So it is also taking part in to expel out the excess water and waste from the cell. Describe the structure of the following and with the help of label diagram: nucleus and centrosome. So nucleus contain all the cellular activities of the cell. It is a spherical in shape. It is composed of the following structure. So if you see the structure of nucleus, then you can see the nuclear membrane. It is a double membrane separating the content of the nucleus from the cytoplasm. So it is a double membrane structure, right? And it is having the double membrane separating the content of the nucleus from the cytoplasm. So double membrane structure, it is having its own compartment, nucleus. Inside the nucleus there is a liquid substance, that is a nucleoplasm, and inside the nucleoplasm the chromosomes or the chromatin materials are present. The narrow space between the two membrane is called as a perinuclear space. So this nuclear membrane is again a double membrane structure, and in between there are some holes, that is called as nuclear pore. Now the two double membrane structure, there is a space in between the two membrane, that is called as perinuclear space. Nuclear membrane has a tiny holes called the nuclear pore. So they are having a tiny pore-like structure. Small holes are there. So they are called as a tiny pore-like structure, right? So nuclear pores. These holes allow the specific substances to be transferred into a cell and out from it. So this nuclear pore are very, very important because they allow the substances to move inside and outside. What are those substances? For example, on the template of DNA is present, that is the chromatin material or the chromosomes. It get opened at the time of transcription, and on the template of this DNA, the RNA are synthesized. Now the RNA has to be transferred out of the nucleus because RNA, these mRNAs are there to form the protein material. And what are the site for the protein synthesis? The site of the protein synthesis is the ribosome. Ribosomes are present in the cytoplasm. So this, through the nuclear pore, these RNA are transferred out of the cell. right? So specific substances has to be transferred into the cell and also out of it. That is the nuclear pore. Nucleoplasm or the nuclear matrix. So what are the substances that present inside the nucleus? That is the nucleoplasm. That is the liquid substance. The liquid substance that is present in between the nuclear membrane and plasma membrane, that is the cytoplasm. And what is present inside the nucleus, the liquid substance, that is called as nucleoplasm or the nuclear matrix. It is a homogeneous granular fluid present inside the nucleus. That is the homogeneous. This is not a heterogeneous. It is a homogeneous granular fluid that is present inside the nucleus. It contains the nucleolus and chromatin. So this is a thread-like structure, okay, along with some protein attached to it, and that is called as chromatin material. Chromatin material made up of DNA and protein. There is nucleolus structures also. Nucleus is a spherical structure that is not bound by any membrane. It is a not membrane bound structure, and it is rich in protein and RNA molecule, and it is the site for the ribosome formation. So at this particular point only, where the nucleolus is present, the ribosomes get synthesized, and this ribosomes, when get synthesized, it is transferred out of this nucleus through nuclear pore. Chromatin is an entangled mass of thread-like structure. It is a long thread-like structure, and it contains the DNA and some basic protein that is called as histones. So these histones are the basic protein, right? And on this histone proteins only, the chromatin material make a wound over it and form the nucleosome-like structure, right? So here you can see a structure of a nucleus. This is a double membrane, nuclear membrane. You can see perinuclear space in between. Then you can see the nuclear pore-like structure. Inside there is a nucleoplasm, that liquid substance that will make the homogeneous ground substance, that is the nucleoplasm. Now in this nucleoplasm only, there are some thread-like structure which is called as chromatin material. At the time of cellular division, this chromatin material get break at a specific point and making the chromosome-like structure. You can see a round bound like structure that is not having any membrane like structure over it, that is called as the nucleolus, and this nucleolus is a site of protein and ribosome formation. Centrosome. Centrosome consists of two cylindrical structure that is called as centrioles. Okay, these are the two cylindrical structures that is called as centrioles. Centrioles lie perpendicular to each other. Now these centrioles, they are lie one above in such a way that one is perpendicular to the other, and each has a cartwheel like organization. Cartwheel like organization. A centriole is made up of microtubule triplets. So 3 microtubules unite together to form the centriole structure, and they are evenly spaced in a ring. So adjacent like this triplet, 9 triplets are formed arranged in a circular form, right? And there is a proteinaceous hub in the central part of the centriole. The hub is connected to the triplets via the radial spokes. So in the center there is a central, that is a hub, and it is connected to the outer triplets by the radial spokes. These centrioles help in organizing the spindle fibers or the astral rays during the cell division. So then it is helping the chromosomes to get divided into the cells which are forms in the future. So they form the basal body of the cilia and flagella. They are also forming the basal body of cilia and flagella. What is a centromere? centromere, and how does the position of centromere from the basis of a classification of the chromosome. Support your answer with the diagram, show the position of centromere on the different types of chromosomes. So here we can see the answer. Centromere is a constriction present on the chromosome where the chromatids are held together. It is a constriction. Constriction means at that particular point the chromosome structure can constrict. Okay, so centromere is a constriction present on the chromosome where the chromatids are held together. Chromosomes are divided into 4 types based on the position of the centromere. So there are 4 different types of centromere, right? So chromosome, and 4 different types of chromosome based on the position of the centromere. So what is the first one? That is the metacentric chromosome. So metacentric chromosome, the chromosome in which the centromere is present in the middle and divide the chromosome into 2 equal arms, is known as metacentric chromosome. So here you can see the centromere is present in the center, and it is the 2 arms of the chromosome are equally at the 2 ends. If you see this, right? So the chromosomes in which the centromere is present in the middle and it divide the chromosome into 2 equal arms, and that is known as metacentric chromosome. Metacentric chromosome. Next one is the submetacentric chromosome. The chromosome in which the centromere is slightly away from the middle region is known as submetacentric chromosome. Okay, so here you can see it is not present in the middle but towards one end, right? So centromere is slightly away from the middle, so it is not dividing the 2 arms of the chromosome equally, right? So this is called a submetacentric chromosome. In this, one arm is slightly longer than the other arm. If you see the structure, this one is a short arm and this one is the long arm. Whereas in the metacentric chromosome, both the arms are of the same size because this centromere is present in the center. But in the submetacentric, it is towards one particular end. So that is the submetacentric chromosome. Acrocentric chromosome. The chromosome in which the centromere is located close to the terminal end, and it is known as acrocentric chromosome. Towards the one end, it is more towards the one end. So it is forming a very short arm and it is forming a very long arm, right? So the chromosome in which the centromere is located close to one of the terminal end, it is known as acrocentric chromosome. And in this, one arm is extremely long. So here you can see extremely long arm and the another one is the extremely short arm, right? So that is the acrocentric chromosome. The last one is the telocentric chromosome. Telocentric. So in the telocentric chromosome, what happened? The chromosome in which the chromosome centromere is located at the terminal end is known as telocentric chromosome. So here you can see the centromere is at the terminal end. So only one side you can see the arms are present. The arms are not present towards the another end, right? So this is the telocentric chromosome. So with that, we have finished the NCERT questions of this particular chapter. We will meet again with a few new chapter. Till then, thank you very much.